so we'll see how far we can get. I have two coupled topics. The first one is for the biologists, the second one is for the physicists. So I'm going to talk first about discovery of a specific new regulatory paradigm for kinesin, and that's going to lead us into heterogeneity towards the end. So I'm not going to go through the movie, but the point was that often motors work in groups, and so it is important to sort of understand, among other things, how the number of active motors is regulated, and we'll see sort of a new idea about that. Okay, so you've heard a lot about motors being important. They're important in neurons, and I won't sort of go through that again. They're important in vesicular transport, but also in invasion by herpes viruses. And so the reason the viruses want to use the neurons to invade is that they can spread through the body without going into the blood, and therefore sort of avoid a lot of the immune system. So it's a very effective way. So in principle, if we understood how the viruses are co-opting the motors, we could block it. Okay. They're also, of course, important for organization and allow the cell to make use of all the different chemical environments and different bounded organelles so that you can deliver things from one to another and, and have different processing in different places. So we'd like to understand how all of that works. Now, one of the things about the cell that's important is not only does transport have to work, but it has to be regulated. So this is an example of a pigment granule cell, and in frogs or fish, they're in the skin. And basically, if you're a frog and you jump up onto a leaf and it's green, you'd like to avoid being eaten. So you may spread out green pigment, and that will help make you invisible. So it's a very good, and these little pigment granules are in general, filled with green pigment. This cell basically isn't fully turning green or black in this case because we've treated it to decrease the number of pigment granules so that we could actually see what was going on. So there they went out. Now, of course, if you're the frog and you jump off of the leaf, then you'd like to be able to turn gray again, and you do that by pulling them into the center of the cell. So this is a regulated process. You need to be able to switch from one to the other. Now, we know a lot about how individual motors work, but we know much less about how this is regulated. In this particular case, it's done by signaling, and we'll get back to that in a moment. So the dominant paradigm overall for motors is that they're auto-inhibited. They're auto-inhibited, which means basically when they're floating around in solution, the tail binds the head and turns off the activity, and then when they get recruited to a cargo, they're active. And so there's a lot of thought that one way to regulate cargo's transport is simply by controlling recruitment. But there's also a lot of evidence for on-cargo control of motor activity. So there, the pigment granules that you just saw basically can change direction, and as far as we can tell from doing a variety of studies, including biochemical studies, the actual number of motors on the cargoes doesn't change. So it's the same amount of kinesin and dynein regardless of whether you're going net minus end or net plus end, and so you're changing something about the activity of the motors rather than the total number. So that's an example of some sort of on-cargo regulation, and in this particular case, there's no recruitment or release. So we'd like to sort of know, how do you do that? Okay, now, the general hypothesis is that you can control transport by signaling, so some signal comes in, and somehow that's going to change the activity of the motors. Now, in this particular case, for the pigment granules, we know that most of this activity is controlled by protein kinase A. So basically, if the protein kinase A is active, you're in the extended state, and if you inhibit the pKa, you're in the aggregated state. And if you start in the aggregated state and you treat with bromocyclic AMP, which is a non-hydrolyzable cyclic AMP analog, which can activate pKa, you go to the expanded state. So this is a case, and you can read about it if you're interested in that paper. So this is a case where we kind of know what the signaling does at the level of the cell, but we're sort of woefully ignorant as far as how the pKa is controlling what's going on. We can quantify sort of the changes of motion of individual pigment granules, and we can see that the travels in one distance change relative to the travels in the other. They're always doing this bidirectional, but you can tune the relative properties, but we don't know downstream what the pKa is controlling. Okay, so this started me thinking that, one of the interesting things is that in disease, signaling is often altered. And so you can imagine that in a disease where, you alter signaling, since signaling can control transport, the altered signaling could control altered transport, which could contribute to disease progression. Also, when you alter the transport, that could alter the signaling, and so you could have various feedbacks. And one of the things that we don't really know about is, in a lot of different diseases, we know signaling pathways are altered, but we don't know how to relate that to disease. So if I tell you in cancer that a particular, signaling pathway is upregulated, and then I want to know, so given that, and maybe that it affects transport, how does that contribute to disease? Really, we can't tell you. So that led to basically a sort of in vitro screen that we wanted to do to look for regulators. So the idea was, let's look at disease-relevant kinases that are altered in diseases where transport might be relevant, and let's see if they seem to do anything. So we looked at junk family and GSK3 beta and CK2, which these didn't give us anything particularly interesting at the single molecule level, but the CK2 did, so I'm going to tell you about that. So CK2 basically is known to be lower activity in Alzheimer's and higher in cancers. And interestingly enough, the amount of CK2 upregulation that you have in cancers seems to be directly correlated with the severity of the cancer. So if you have, the more CK2 you have, the worse off your prognosis. Okay, so that brings me into how we're going to do the single molecule tests. So let me just remind you that if you've got sort of in vitro, you have a cargo and you can put molecular motors on it and you have a microtubule, what you can look at is distance, the sort of how far the cargoes go. You can look at how fast they go and how much force they produce. Those are sort of the typical things we look at. And we do this with an optical trap. Remember, if we attach a bead to an optical trap and then we basically let it bind, if we turn off the optical trap, then we can just watch how far the cargo goes before it falls off the microtubule, and that allows us to measure travel distances or processivity. Here's what that looks like where we have a single kinesin on the bead. Goes a little bit of distance and falls off. So you can basically place the cargo on, let it walk, and see how far it goes. Now we can also measure force doing the same thing. There we just leave the optical trap on, and because there's it's a harmonic potential, by looking at where the motor stalls, basically we can measure the force that's produced. And you've already heard about using optical traps to measure forces. You've already gotten the idea from a few other talks that forces add. We were actually, I think, the first lab to show that forces in molecular motors are additive. That is to say, the peak force is additive. I think there's still some debate about the frequency with which forces add when you have, say, two motors, and we can discuss that later if people are interested. Okay, so in principle, by measuring force, you can count the number of motors. Okay, so this is one motor traces for kinesin, just to show you what the data looks like. Okay, so what you can do then is you can do an experiment where you take some recombinant kinesin and you treat it with CK2, and then you recruit it to the bead, and basically you do this either untreated kinesin or CK2 treated, same amount of kinesin, and you look at how far the beads go. And so in the untreated case, basically they're going about 2.2 microns. So this is, to do these experiments, to make them fast and easy, we chose to work in a binding fraction of around 1. So this is a sort of low multiple motor regime. A single motor would have a processivity on the order of 1 micron. So we're in the multiple motor, but not massively multiple motor regime. And what we saw when we did exactly the same experiment with kinesin that was treated with CK2 was that the travel distance was at least double. So there's this, well, bar here at 8 microns, it's not that they went 8 microns and fell off, it's that they moved out of the field of view and we stopped watching them. So this 4.3 microns is an underestimate of the travel. Okay, and this all basically was published in this Nature Communications paper in 2012. Yes? Yes, we're treating the motor before it's on the bead. And so that will lead to various possible interpretations. We'll get back to that. I should mention, by the way, that although we are treating the motor before we put it on the bead, the way we're attaching it to the bead is via an antibody-specific linkage. It's an engineered linkage. So any sort of charge probably isn't affecting the recruitment, although we tested that, and we see no difference in the number of motors that are recruited to the bead as a function of the activity of the kinase. Any other? Okay. So, possible interpretations, in addition to sort of more motors being recruited to the bead, is that maybe this phosphorylation via the CK2 is increasing processivity of the motor, or maybe somehow there are more motors that are active. So, because we know that if you have more motors that are active, you go further distances in general. Of course, you also have higher forces. So, how can we test that? Well, one thing we can do is we can look at forces. And for the CK2 kinesin beads, we saw basically a distribution of forces that looked like this. And this basically is the hallmark of having two motors that are active. So the basically here, because this was very sort of in the very, very low two-motor regime, we basically saw just the single motor force. Okay. So that suggests that we have more motors that are active. Doesn't say that we don't have increased processivity. To test that, if you think that you have more motors that are active, you just have to do more of a dilution, right? So if you do more of a dilution, if you basically, I don't remember exactly how much the dilution was, but then you can get down to the 30% binding fraction where you're really looking at single molecule properties. And what you discover is that the single molecule properties of the wild type kinesin that's untreated and the kinesin that was treated by CK2 are identical. So it looks like from this that the CK2 is somehow changing the number of active motors. So that was sort of a surprise. And so you can ask basically, is this an in vitro artifact or is it relevant for in vivo? And you can ask sort of also what is the nature of the effect. So what you can do is, you go to our friendly lipid droplet transport in the cell and you look at sort of basically you fix the strength of your optical trap and you set the optical trap to be sort of moderate strength so that about 90% of your lipid droplets escape from the optical trap when you grab them. And then using that same strength optical trap, you look at how many lipid droplets escape when you do RNA interference and knock down CK2. And what you discover is that using two different RNAis, the probability goes down a lot of escape. So that's basically just a measure of efficacy of force production. So basically, that tells you two things that are interesting. One is, it tells you that because we do the control with like a scramble RNA and we don't see this, it's really because you're knocking down CK2. So that tells you first that CK2 looks like it is controlling the number of active motors in vivo, and that without CK2 activity you have fewer. It also speaks to this question of how well the kinesin 2 is doing together, or the kinesin motors are doing together, because it looks clear that you're getting additivity of forces because you're able to escape better. So this suggests that the motors are working well together here, and that you have multiple motors that are moving the wild type ones. Now I should stress that, as you'll see, that this just says that some kinesins forces are pretty nicely additive. It does not, we have not proven that the lipid droplets are moved only by kinesin 1, and so, and as I'm going to show you later, this regulatory pathway seems like it controls basically all kinesins. So what this is telling you is that there is some kinesin on the lipid droplets, and you're having fewer of those guys active, and then forces are no longer as high. So it says that you can get good additivity of forces for some kinesin, but it doesn't say kinesin 1. Any questions before we go on? Yep. I can't eliminate that as a possibility. Well, in vivo, I can't eliminate that as a possibility. I tend to think that, Occam's razor is pretty good. So Occam's razor tends to say that if you see something in vitro, it probably is the same as what's going on in vivo. In vitro, what I can tell you, as I'll show you next, is that we really are changing the number of active motors. So my belief is that the same thing is happening in vivo, but, there's always surprises when you go in vivo, so it could be different. Yes? In my in vitro experiments. You mean, why is it that we get additivity of forces? Okay, so we think that basically we are getting additivity of forces because both motors are bound and they're both supporting load simultaneously. However, I need to be clear about this experiment because this is what we're doing is basically sort of a crude experiment in the sense that we've got a cargo and we have an unknown number of motors on this cargo. When we go to the binding fraction of 1+, we have multiple motors on here. And so what we're looking at basically is the probability that when this thing comes down, you end up having 2 motors engaged or 3 motors engaged, okay? But we could have hundreds of motors on here and in general just see that 2 are engaged. Right. So it's not like a clean 2-motor experiment where you only have 2 motors and you're asking, do the 2 motors. So when we see the higher forces, yes, it's it's clear that both of them can walk, both of them can produce forces, but it could be that you need. Well, no. So for the precessive motors, as long as the velocity is the same, there's no issue. They can sort of walk, because what controls travel distance is basically when one detaches, the second guy keeps walking, and if the first guy can rebind before the second guy detaches, then you go very long distances. It turns out that isn't so much of an issue because for kinesin 1, the rate of detachment under forward load is much more rapid than the rate under backward load. So if one starts to act as a brake for the other guy, he gets pulled off. And that's actually addressed in sort of experimentally, it was determined by Bloch, but sort of simulations showing that are in the heterogeneity paper that I'll talk about next. Okay. And it was also done by Michael Deal and you. So this is sort of the notion that this sort of sensitive dependence under forward load allows motors to avoid getting in each other's way is, I think, pretty accepted at this point. Okay, so CK2 is relevant in vivo and in vitro, and so now the question, well, how is it working? So in principle, it could be changing motor activity, either on rate or the overall number of active motors, or it could be recruitment of motors to the cargo. And so we wanted to address that. And so the first thing you can do in vitro is you can get rid of the cargos. Right? So what we do is a simple pelleting experiment to look at activity, where we take microtubules and we add kinesin to them, and we're using AMP-PMP, which is a non-hydrolyzable analog, because the AMP-PMP locks kinesin in the ATP-bound state, which is a high-affinity state for the microtubule. So in principle, all the active kinesins should bind to the microtubules, and you pellet the microtubules, and then you look. So in the absence of CK2, you see some kinesin coming down, and in the presence of CK2, you see a lot more coming down. Okay, and okay, so it turns out one of the sort of surprising things about this is that the kinase activity of the CK2 is irrelevant, which took us years to figure out. So TBCA is a specific CK2 inhibitor, blocks kinase activity. It basically, the presence of CK2 and TBCA has exactly the same effect as CK2 without TBCA. So the presence of CK2 is important, but the kinase activity is not. Okay, so basically the amount of kinesin that comes down is affected by CK2. Now, one thing that's important here is that in this particular experiment, well, let me go on it. Okay, so there's no cargo there, and that says that it's probably not due to additional recruitment of motors to the cargo that matters. Okay, so models could be that CK2 is releasing the head-tail inhibition. CK2 could function as a crowding agent, preventing denaturation of the protein. It could be phosphorylating kinesin, and I just sort of said that it's not, and we'll give you a little bit of additional evidence there, or there could be something more interesting. Okay, so the first experiments were done with full-length kinesin that had a tail, and that was basically, you got more kinesin on the microtubules for the full length, but you can make a truncated version, K560, that has no tail, so it's always active, and that basically was also activated. In fact, this guy was activated more than the wild type, and we think that's because the tail basically, the presence of the tail slows the transition that I'm going to tell you about into the inactive state. So these guys go inactive more faster than these guys. And so then the CK2 does more as far as reactivating them. Okay, so that was independent of the tail. So could it be denaturation? Well, you can take various proteins that you can use as crowding agents like BSA or another kinase, GSK3 beta, and you can use the same quantity of those, and you don't see the same effect on the kinesin activity as you do from the CK2. And in fact, CK2 alpha prime and CK2 alpha are very, very similar, but they're slightly different, and CK2 alpha has a much bigger effect than CK2 alpha prime, suggesting that this is specific and it's not simple crowding. Okay. So it's independent of kinase activity. I told you about the TBCA experiments. We actually went and mapped the site, which is 520, that was being phosphorylated by CK2. We made a point mutant that wasn't phosphorylatable. That still went inactive, and it was still reactivated by CK2. I guess so. Yeah, so this one basically has, this is just showing these are radioactive incorporation to show the sort of the amount of hot phosphate that's incorporated in the presence of CK2. So CK2 autophosphorylates, and then you can see here that the kinesin is being phosphorylated, but the 520A, most of the phosphorylation is eliminated. Here basically is the pull down, the microtubule pelleting experiment without CK2 and with CK2, you can see that they reactivate. Okay, so it's kinase activity seems like it's a red herring. Okay, so that brings us to mechanism. Any questions before we? So CK2 is a monster. It's a tetramer of an alpha and an alpha prime and two beta subunits. And I think there are structures for alpha and alpha prime, and I don't know if there's a structure for the tetramer, but we don't know what to do with it. So I think this is a problem that's crying out for theoretical analysis, but we'll get, yes? We do not know. No, it would be great to know, but we don't know. Okay, so mechanism of regulation. So if you think about kinesin basically has different enzymatic states, and they have different affinities for the microtubule. So our model for active or inactive motors is basically whether they can bind microtubules. So my first model was that, Ron Vale a long time ago had speculated about the similarities between kinesin and G proteins, and I thought, here's a case where kinesin is really acting as a G protein. It's stuck in the weak affinity state, and CK2 is acting as an exchange factor and is popping it back. I was certain that this was what was going on. Of course, didn't work. But so how do you explore that idea that basically you're stuck in the ADP-bound state and that CK2 is acting as an exchange factor? So one thing you can do, okay, there's just a model that the head is trapped in the ADP-bound state, causing low affinity. Okay. So one thing you can do is, you know that when the motor is floating around in solution, it's in the ADP-bound state. So you can take hot ATP, where it's the second residue of, so you have ADP, which is APP, or ATP is triphosphate. You can have this guy be P32 labeled, so that when the kinesin binds and hydrolyzes the phosphate and then floats around as an ADP-bound guy, it's radioactive. And so then you can detect whether the kinesin is bound to ADP or not. And so in the absence of, so when we do this, and then we look at the kinesin via a dot blot experiment, so we're just putting a dot of kinesin, the kinesin sticks to the filter paper, you wash it off, so basically any a hot ATP or ADP in solution that isn't attached to the kinesin washes away, and you can see basically radioactivity. In the presence of CK2, you see much less radioactivity. So that suggests that the presence of CK2 is releasing ADP from the kinesin. So that was great. I was all thrilled that we've solved it. Unfortunately, you can do a few other experiments, right? So one thing you can say is, if this is right, it shouldn't be able to go inactive if it doesn't have ADP. So you can actually purify kinesin if you work hard. You can purify kinesin in the apo state, in the null state, and you can confirm that it's in the null state, because in the null state, if you take your kinesin and you incubate it with your microtubules, they just will pellet without any AMP, PMP. And when you did that, you could confirm that it was in the null state, but if you age it some, what you discover is that without CK2, you get a very low amount of the motor coming down, and when you treat with CK2, you get all of it coming down, and so CK2 is able to activate the motor even when the motor doesn't have ADP bound. So that was a little disappointing. You can also do the following experiment. You can do a sort of pulse chase experiment. So you take your kinesin, and first you incubate it with this hot ATP, which is converted to hot ADP and sits there. And then you can, after incubating it some, you chase with a lot of unlabeled ATP. And so the idea is, if the motor is holding on to the ADP and is not releasing it, when you give it new ATP, it's not going to release. So basically, when you do this pulse chase experiment, you first load it up and you confirm that it's pretty radioactive, and then you do a chase where you chase with unlabeled ATP. You see that the radioactivity goes down. If you do that same experiment, you load them up with ATP, hot ATP, and you wait 4 hours and they're inactive, and now you do your chase at 4 hours with unlabeled ATP, you see basically that radioactivity goes down, so it's able to exchange the bound ADP for a new ATP just fine. So all of these things suggested that it's not, unfortunately, stuck in the ADP-bound state. Okay. So the next idea was, it turns out that CK2 actually binds the microtubules directly, and so maybe CK2 is making a link between the motor and the microtubule and just holding it there independent of everything. Maybe it's not really that it's affecting kinesin's activity per se. So if that were true, so if kinesin's binding to the microtubule is important, if you basically put in more and more ADP, you can compete the kinesin off of the microtubule, if only its enzymatic activity binding to the microtubule is important. So what we see is that as we add increasing amounts of ADP, we get less and less kinesin bound, but the amount of CK2 on the microtubule stays the same. So that suggests that it really is that CK2 is changing the amount of active motor, and it's not due to sort of it being linked to the microtubule per se. Okay, so the conclusion from all this is that it's causing some sort of conformational change to release the ADP, but that nucleotide release is secondary, and it's probably not why it's inactive. So it leads to the question of why is the motor inactive? And I only have a partial answer. Okay, so the answer is the following. So I started thinking about this, and at a conference, talking to Michio Tomoshige and others, we were sort of talking about the conformational changes that kinesin goes through. And when you think about kinesin as a machine, there are actually coupled conformational changes. So you have something that happens at the nucleotide binding site, and that causes a conformation in the microtubule binding site, but also in the neck linker. So I thought, what if this inactive state is some sort of mixed conformation where, you're bound to ADP here, but maybe the rest of the motor doesn't know it, or it's some sort of mixed conformation that you wouldn't usually see. So you can probe that by looking at the relationship of the neck linker to the head. So usually the neck linker is docked to the head, like drawn here, in the ATP configuration, and it's undocked in the ADP configuration. So we decided to look at the neck linker and see whether the details of the neck linker changed over time. So this was with Michio Tomoshige, who had fret pairs, and basically, if the neck linker is docked, then you should have high fret, and if it's undocked, you should have low fret. And what we saw is that if we took the active kinesin, we basically saw that it was in the ADP-bound state, and in the ADP-bound state, you'd expect the neck linker to be undocked, and that's what we saw at t equals 0. As we aged it, and the kinesin went inactive, we saw that it looked like the neck linker was docking with the head. So that was interesting. So it seemed, and the amount of increase of the percentage of guys that were in the docked state was consistent with the percentage of motors that were inactive. So that led to this idea that maybe sort of being in the docked conformation is somehow associated with being inactive. So if you basically let it go inactive and you treat with CK2, you can reverse the conformational docking. So that was encouraging. So we think, so basically if you just take kinesin and you age it in the presence of CK2, you don't get this rise. If you let it, the docked state go up and you treat with CK2, you revert back to, almost the undocked state. So it looks like inactivation is associated with neck linker docking, and this is certainly going to be tell us something. We don't know why neck linker docking is associated with inactivation, and so I think that's where we need some help from a theorist. Okay, so that's kind of what I have to tell you about mechanism, and we need to understand that. And so I want to briefly tell you that what I've just told you is relevant for other kinesins, because it leads to a really interesting model of how you could regulate things. And then we're going to sort of talk about heterogeneity. So the kinesin head is highly conserved, and so you might think that maybe this mechanism carries over. So, we had done this with kinesin 1. We decided basically to look at things that were pretty diverse. So we looked at SEMP-E, which is way down here, and is a mitotic kinesin. We looked at EG5, which is a mitotic kinesin, and we looked at kinesin 2, KIF3, which often functions in interflagellar transport. And what we saw is that each one of them went inactive over time. So this is the SEMP-E, this is the EG5, this is the kinesin 1, and this is the kinesin 2, and with considerably different T1 halves, considerably different inactivation rates. And each of them could be reactivated by CK2 treatment in vitro. So it looks like it might be sort of a general phenomenon. Now, what's interesting about this, I think, is when you look back at this data, you'll realize that these two are the mitotic guys, and both of these are sort of vesicular transport. Now, you might think from a biological point of view that, if you're a mitotic motor, you really want to be active in a narrow window of time and not in other times, right? So from a design point of view, you could make an argument that wouldn't it be cool if you could have your motors be inactive and waiting and then reactivate them when and where you want them. So by localizing CK2 to particular places, you could localize motor activity to where you want it. Interestingly enough, during mitosis, CK2 is reported to heavily decorate the spindle microtubules. So that would be a very nice way of locally reactivating EG5 and SEMPI and things. Yes? This is basically taking the motors and doing microtubule pull downs and looking at how much comes down as a function of aging time. Yeah. Is all in vitro. Aging on ice, I think. Yeah. So we're just, aging under identical conditions on ice in a test tube. So in these cases, the first thing we do is I think we do an initial pull down and then release steps so that we're starting with all active motor. So at t equals 0, everything's active. And then we basically age and we look at how much. Yeah. And then doing it again, basically. Yeah. And quantifying the percentage that's coming down. Okay. So anyway, it leads to a very intriguing model for regulation. Whether or not that ultimately proves to be the case or not remains to be seen, but I've at least shown you that for kinesin 1, decreasing CK2 amounts does lead to changes in force production. Okay. So I want to return for a moment to this FRET study. Okay. So one really interesting thing about this FRET study is that the inactive state is a high FRET state, and these two FRET probes are 215 and 342. They're reasonably, far away from each other in nucleotide sequence. And what that means, because you have a high FRET efficiency, is that it's not a disordered state. If you calculate what you would expect from that number of nucleotides, basically you wouldn't get high FRET. So the fact that when it goes inactive, you're getting a high FRET state means that it's not an unfolded state. Okay? And I think that's important, because what this is saying is that, and the fact that CK2 can reactivate also, I guess CK2 could be acting as a chaperone, but this suggests that it's not. So what that suggests from a protein folding point of view is that the low energy state, which is the state you're getting when you wait a while, that ground state is not the active state, because the ground state, it's inactive. Okay? And so what it suggests is that the active state of kinesin is an intermediate before you've gotten to the ground low energy state. Okay? And so that's really interesting, because, there's this notion that when you fold, you basically funnel down to a unique conformation, a folded conformation. And we don't have any issue with that, but because this is an intermediate, it suggests that maybe you could have multiple different conformations, right? So it leads to the possibility that if you have different intermediate conformations that are active, you could have heterogeneity in function. And it doesn't contradict sort of the simplest notions of protein folding having a unique folded state. Okay. So that sets up the second part of the talk, which is about heterogeneity and the question of, is there only a single intermediate or not? Yes? No. So the CK2 is binding on the empty track, but the CK2 also interacts with the kinesin directly. And one of the things I didn't stress is that using the FRET, the neck linker docking, you can see that single motor domains, single heads go inactive, and single motor domains can be reactivated by CK2 in solution without any microtubules around. So the inactivation and reactivation is, we think, independent of the microtubules, although it is true that CK2 localizes to the microtubules when they're present. when CK2 binds, so most of the time when the motor is around, when CK2 binds, it causes release of the ATP. So there is an associated chemical change. And when we do tests, even without the nucleotide, to do those tests, basically, the only way we can test for activity is to look for the ability to walk on the microtubule, and then we have ATP present. So it may be that to get the CK2, once the CK2 induces a conformational change, to get it off may require a chemical change. That would be my guess, because I don't believe that energy comes from nowhere. Yes? I think it is some sort of allosteric, conformational change, but we don't know the details of how it's happening. Okay, so that brings us to heterogeneity, and I can slow down now. I apologize for the speed of the first part, but I wanted to get through it all. Okay, so, motors, you can get to the single motor limit pretty easily by attaching motors to beads and using fewer and fewer motors in solution to get a smaller binding fraction. So if you go to beads where the probability of a bead binding when it's put into contact with the microtubule is about 10 or 20%, you're pretty sure that you're at the single molecule level. And so what we see when we do that is that we see different velocities. So these individual tracks, these are individual tracks of motors walking along microtubules in vitro. And we see some, with a velocity of 350 nanometers a second, and some, moving at 800 nanometers a second. And so there's some clear heterogeneity here as far as the velocities. And now you can ask, well, is this really, and so I would classify this as functional heterogeneity, but of course, first question that you'll have to ask is, well, are these really identical? And, you can imagine all sorts of scenarios. So I'm going to walk you through a bunch of scenarios and eliminate them one by one. Yes? Oh, this is just the number of tracks that were in this portion. So basically, we're seeing most of them in this particular small n experiment. Most of them were going at around 800 nanometers a second, and there were 3 or 4 that were going slower. So it's just sort of an indication of relative amounts. Okay. Okay, so, yes. So we chose, basically, some of these are really long, most of them are actually shorter, and there are a bunch in here that you can't really see. This is not under any load. We put them onto the microtubule, we turn off the optical trap, we let them go. And the distribution of travel distances basically gives you a decaying exponential with a mean travel of around 1, consistent with single motors. Okay. Yeah. Oh, this is saturating ATP, like, this is, I think, 1 millimolar ATP. I don't think we've done it. I don't think we've done it. We've done some other perturbations we'll talk about, but I don't think we've done that one, so I can't answer it. Okay, so it was surprising to us that we saw these different velocities. And so possible explanations. Okay, one is that the differences are due to basically stochastic variation. And if you think for 2 minutes, you'll realize that can't possibly be true because the law of large numbers says that things should converge, and those are clearly not converging, but I'll show you a simulation that supports that. These are single motors for the moment. All are done, and I'm going to show you true single motor where they're GFP tagged and it's only single motor. So these are single motors. These are not multiple motors. Yes? So to the extent that we can look by grabbing a bead and looking again, slow guys are slow and fast guys are fast. We don't see, in the really low motor limit where we're pretty sure there's only 1 motor on the bead, we haven't seen any conversions. Seems like. Or at least the transition, if it occurs, is over a time scale that is different than the experimental times that we look at it. Okay. Okay, so there could be different chemical changes due to purification. It might not happen in vivo. They might not be identical. They could be post-translationally modified. They might have different amounts of head-tail interaction since the tail is auto-inhibitory. They could be moving along different microtubules with different amounts of post-translational modifications. There could be surface effects because you have your microtubule that's sitting on your surface, and so you could be moving along the top protofilament versus the side one. There could be differences in how the motors are attached to these artificial cargoes, or there could be something really interesting like maybe different fold fates. So let's go through them all. Simulated motors, basically if you do a stochastic stepper that has the right velocity, you see this kind of distribution. And the tails with the low velocity or the high velocity are the short guys, basically, because the law of large numbers says that on average, if you have a random variation in your each cycle, if you put lots of cycles together, you get a pretty well-defined velocity. So all the long ones have a pretty well-defined mean, and you get spread as they're shorter. So that's what you'd expect due to stochastic variation. It's not what we see. So let's eliminate that. So what about chemical changes due to purification? Maybe it doesn't happen in vivo. So we teamed up with I think Marvin Tannenbaum, I think. So he had been a postdoc with Vail, and they put in kinesins into cells which were multiply tagged but were truncated, so they're not attached to cargoes. So they're just kinesins walking around, and they had only tracked a small number, so we got the movies and tracked lots. And basically, let me show you the movies here. If you look at these guys walking, you'll see some move pretty fast and some move much slower. So that the green ones are faster and the red guys are slower. You can see it's going slow. Okay, so we do see some heterogeneity in vivo. In fact, what you can do is you can track everything, and then basically, and we got like 1,200 tracks or so, and then we can select the good ones. And by good ones, what we did is we wanted to do this objectively, but we wanted them to have a well-defined velocity. So what we did is we fit to lines and looked for the correlation, and basically if they were quite well-defined velocities, high correlation coefficient, we took those, and anything which had pauses or looked like they got stuck or anything, we tossed. Okay, but this was in an automated fashion based on a correlation coefficient. There was no sort of eyeball transform of tossing the slow ones that we didn't like or anything. And what you see is a distribution like this, where there's this long tail that goes towards zero. So this could be sort of decomposed roughly into two different populations. And, one had a velocity of, 1.2 microns per second and a, decay length of 0.8. And the other was, 0.6 microns and a decay length of 2. I'm not sure exactly how statistically significantly different they are, but it's not like these guys are super ill or anything. So it looks like there's heterogeneity in vivo. Okay, now, people have seen heterogeneity in vivo before, and the explanation is always, well, you have post-translational modifications of the motors and post-translational modifications of the microtubules, so it's probably due to that. So all this does is say that there is heterogeneity. It doesn't say the source of it. And I think you have to go back to in vitro studies to get a cleaner idea of what's going on. So I've just told you that okay, so we did this. It does happen in vivo. Now we're going to sort of address a number together. So we're going to look at motors that are made in E. coli. So they are presumably not post-translationally modified because kinesin is not normally made in E. coli. And they're not going to have different amounts of head-tail interactions because we're going to make K560, which has no tail. And because we're making the microtubules in vitro, the microtubules presumably, if they do have any post-translational modifications, it's all random. Okay, so here's what we see. These were K560 labeled with quantum dots. We see a pretty wide range of velocities. Some of them were quite long. There are lots that are short. And if you look at the distribution, it's consistent with single molecule behavior. So far, so good. So what about surface effects? Moving along the top protofilament versus the lower protofilament. So there, you can raise your microtubules off the surface and basically examine motion, and you can quantify the details of sort of how we figured out how far away from the surface they are is in the paper, and I'm not going to go through it. But anyway, we still see heterogeneity. We don't have that many tracks because they're hard experiments, but they're still heterogeneous, so it's not a surface effect. Okay, so what about how they're attached to the artificial cargos? Now, we had done this with quantum dots, where we had engineered the linkage, the K560's his tags, and we thought that that was good enough, but the reviewers disagreed. So we made K560 GFP, which has no cargo and should all be identical. They were made in E. coli again, and basically imaged them in turf, and again, you see the same thing. And we can get lots of data because it's data in parallel, so you see a bunch of these sort of slow guys, as well as sort of a well-defined, peak at around 600 nanometers per second or so. Okay, and that, in this particular case, it can be, that distribution can be fit by the sum of two Gaussians with sort of, velocities centered at like, 800 or 900 and 600 or something. Okay, so that led us to the notion that maybe we are really looking at motor heterogeneity due to folding. And so because we don't know the details of the heterogeneous state, the question is, how do you prove that? And so Bernie Brooks, who I interact with frequently at Kios in Seoul, suggested that, well, if you think about it from a protein folding point of view, if you change pH, if these are two different folded states, they should, potentially have differential sensitivity to pH. So maybe you can alter the percentage that's in one state or the other by changing pH. So we did that with this GFP-K560 turf experiment. And what we see is that at 5.5, we basically have a much narrower range, and that the distribution looks much more Gaussian. So, at pH of 7, which is where we had been looking, we had this long tail, but at 5.5, they were pretty nicely clustered. So we can definitely change the percentage of slow versus fast guys. And if you go to higher pH, it's even more spread out. So you can quantify, so basically roughly 100% of them are all the same Gaussian at 5.5. It's about 50-50 at pH of 7, and it changes. Okay, so you can also do things with protein folding. You can sort of do crowding. You can do temperature. They give you similar results where you can switch around the percentage that's in the two different populations with these perturbations. So we think basically that there are a couple of different folded states. I'm not saying that it necessarily has to be two, but I'm saying there's more than one. Okay, so let me end there. I first told you about a novel form of kinesin regulation where the kinesins go inactive and can be reactivated by CK2, and I told you that that's true for all the kinesins we've looked at. And because the inactivation involves sort of going into the lowest energy state, which seems to be highly ordered as indicated by the fret, that seems to be the low energy folded state, and so the functional state is not the low energy state. So because it's an intermediate, it sets up the possibility that there could be different folded intermediates, and that may be consistent with sort of this observed functional heterogeneity. So let me end by suggesting that we're desperately in need of theoretical input for sort of why neck linker docking leads to inactivation and sort of the underlying ramifications of heterogeneity. And my suspicion is that at a functional level, it's because you have slightly different conformational changes around the ADP sensing, but that's speculation. So we need sort of to understand these better. And the people who really did the work, let's see, so all the recent heterogeneity was done by Babu, and a bunch of other people in my lab contributed, as well as the Tomashige lab, and some Steve Rosenfeld, And so I'd like to thank you for your attention and mention that I will have an open postdoctoral position, likely, if there's a student in the audience who wants to talk to me. Thanks. So I don't know whether CK2 is acting as a chaperone or not. I haven't tried putting in other ones. I mean, originally I was thinking that it was acting as a chaperone, but then my enthusiasm for that idea decreased when I found out that the inactive state seemed highly ordered, right? As judged by the Fret pair. So
chaperones promote certain conformation over other.
Right. So, I mean, certainly CK2 in that sense is acting as a chaperone because it's promoting one conformation over another. Whether other chaperones can do the same thing, I don't know. It would be interesting to find out.
Yeah. Steve.
Yes.
So if I look from a broader perspective, the motor gets activated when its ATPase domains are ready to perform their ATPase activities in a way. So this is somewhat similar. For example, if you have the DNA and the promoter site is not accessible to the transcription factors. Mm-hmm. So is there some kind of an analogy one can draw with both this?
Yeah, so I think, you know, when I think about this, I don't know whether you can draw an analogy. I think, you know, I've thought a little bit about why you might have this kind of regulation as well as, say, head-tail regulation. And my suggestion is that they're possibly relevant for different cases. So, you know, when you think about the traffic jams that we're observing, you know, that India's observing, you could imagine that those are quite long-lived. And maybe basically, if they're very long-lived, you can't get rid of them. But maybe if the motors in the center of that go inactive, maybe you can resolve them because stuff can push you out of the way, right? So you could imagine that maybe you don't want your motors to last too long because it gives you sort of some problem. Now, we also don't know anything about how activity of the motors themselves affects their inactivation rate, right? The inactivation we're measuring is in solution with no microtubules around. And so maybe the active guys that are walking never go inactive until they basically fall off and are not able to walk. So I think there's really interesting questions about, you know, when you have these multiple levels of regulation, when you use them and why, and I don't think we know. Yes?
I mean, your neck linker docking for inactivation, that might be a candidate for one other conformation that you have. So did you try to somehow sort the slow and fast ones and do the Fret experiments with those?
No. That's an interesting, no, we haven't. And that would be interesting to do.
Provided the inactive form is not completely inactive, which,
right,
I don't know, but it could, I mean, if the fraction of time it stays in this docked state changes.
Yes, I mean, you're right that you might be sort of the slow guys. So it would be interesting to look, basically, I think what you could do pretty easily would be to look at the evolution of the velocity distribution over time as you go inactive, right? So if you basically start at t equals 0, where you know that they're all active, and then watch them as they go inactive, if you saw a shift towards the lower velocity, you know, the proportion, that would be consistent. So that's a really good experiment to try. We've never tried it. Yes? Oh, and let me suggest that I'm happy to take questions, but we only have an hour for lunch, so go ahead, but...
Yeah. I think we should stop here.
Okay, I'll answer your question after.
So this is related to the multiple intermediates that you're talking about. That's very interesting. So do you have any sense of the barriers between these intermediates? And it has a signature of IDP kind of behavior where it has a flatter basins, I guess.
So we don't have any idea about the barriers between. What I can tell you is that they must be somewhat high because we don't see, we've looked at a lot of individual trajectories and we don't see, while they're walking on the order of, a second or 2 seconds or 3 seconds, we don't see a change in velocity from slow to fast or fast to slow. So we don't see interconversions, at least while they're walking. And when we've done very, very limited experiments on looking at the same motor, taking 5 or 10 attempts on the microtubule, the velocities are always slow or always fast. So we haven't seen the interconversions, and we don't know.
I think I will end this. You can catch.